my name is Hannah Smith. Here is a roadmap of my research project. The overall story of this project is covalently bonding a protein to a solid support called resin. This project involved a specific protein called carboxylesterase EST1 from hypothermophilic bacteria. Hypothermophilic means the bacteria and its proteins are stable at high temperatures. The actual full name is a mouthful, so we just call the bacteria SSO and refer to the protein as SSO protein. The first step in this project was producing protein that could be immobilized. We wanted to form a covalent bond between protein and resin. I'll talk a bit more about this covalent bonding later, but essentially we needed specific groupings of atoms in the protein to properly form a covalent bond. This is where unnatural amino acids come in. The human body uses 20 natural amino acids, and these amino acids, they make up proteins, so different amino acids give proteins different functions. However, Professor Young's lab has developed a way to use amino acids that are outside of that natural 20. These special amino acids are called unnatural amino acids. Adding unnatural amino acids into a protein changes the protein's properties and functionality. By adding certain unnatural amino acids, we make it possible to covalently bond proteins to other molecules, like resin. This slide shows a natural amino acid, tyrosine, and also an unnatural amino acid, PPRF, that's created from tyrosine. The unnatural amino acid has a triple bond that's going to be important for immobilization. So basically, to summarize, we put an unnatural amino acid in the SSO protein to allow it to covalently bond to resin. After producing what we thought was SSO protein, we needed to check if the protein was actually present and active. You can't just tell by looking at a vial whether SSO is present, so we used a color change assay. Essentially, we put the SSO protein together with a compound that turns yellow when the SSO protein comes and cuts that compound up. So if we saw a color change, that meant that the SSO protein was present in cutting up that compound. If the protein was present and active, then we could immobilize it by covalently bonding it to resin. One of the covalent bonding methods that we used is called Glaser-Hay. Professor Young's lab developed a method of using Glaser-Hay on proteins. I have shown some important aspects of the molecules, but the circles basically represent everything else. It's too messy to show the whole chemical structures here, but essentially these two triple bonds will react and connect with each other. The result of the reaction is an immobilized protein that's covalently bonded to resin. So, what's the purpose? What's the point of this research? Why bond proteins to resin? The point of bonding SSO protein to resin was to develop a procedure for protein immobilization that could be used for all kinds of different proteins. Different proteins do all kinds of different things, and some are super useful. Many proteins together can even be used to detect a bunch of diseases. Being able to use proteins in research is important, but proteins are also fragile. In the wrong conditions, proteins denature and they stop working. One big benefit of bonding a protein to resin is that it creates more stability for the protein. The resin is kind of like an anchor holding the protein together. Immobilization means that researchers can use harsher conditions on proteins, and therefore they can do more experiments with a wider range of chemicals. Another benefit of immobilization is recyclability. Some proteins take a lot of time and money to generate. When proteins immobilize to a resin, it can be reused more than once. Usually protein's just floating around in solution, and so if you change out the solution, you're going to lose the protein. When protein's on a resin, you can change out the solution and the resin's going to stay at the bottom with the protein attached. Then you can run multiple experiments without having to make new protein, which saves time, saves money, saves resources. A long-term future application is the development of a protein biochip. So many proteins could be attached to a support to form a sort of biochip, and it would be a cluster of many proteins that could be used to detect diseases very rapidly and effectively. Proteins are really small, and so the protein biochip would also be able to be small. So it could be transported to and used in areas that lack good medical infrastructure. And obviously having a small portable tool to detect many diseases at once would be great. So I hope you see why this research is important and has potential for a big impact. Thanks.